Most of us think about inheritance in a simple way. Parents have kids, the parents pass their genes down to their kids. You get your mom's eyes or your dad's nose. This is how it works for people, for dogs, for trees. It's a straight line from one generation to the next. Scientists call this vertical gene transfer. The family tree makes sense. It's a tree, with branches splitting off, but the information always flows downwards, from parent to child. But that's not the whole story. There's another way to get new genes, a weirder way. It's called horizontal gene transfer. Imagine you could get a new skill not by learning it, but by just touching someone who already has it. You shake hands with a musician and suddenly you know how to play the guitar, or you borrow a book from a friend and by holding it you absorb all its knowledge. That's what horizontal gene transfer is like for single-celled organisms. It's the transfer of genetic material between organisms that are not parent and child. It's sideways. It's a friend giving a friend a gene, a neighbor sharing a genetic secret with a neighbor. This process is a huge deal in the world of microbes. For bacteria and other tiny life forms, it's a primary way they evolve. They don't have to wait for generations for a useful mutation to pop up randomly. They can just grab a useful gene from someone else who already has it. This makes them incredibly adaptable. It's like a software update they can download from the internet instead of waiting for the manufacturer to release a new model. And this isn't a rare freaky event. It happens all the time. It's a fundamental part of how microbial life works. It scrambles the simple family tree into something more like a messy, interconnected web. So, how does it actually happen? How do these tiny organisms share their genetic code? There are a few main ways they do it. Think of them as different methods of sharing a recipe. The first method is called transformation. This is maybe the simplest way. Sometimes a bacterium dies. When it dies, its cell wall breaks apart and its DNA spills out into the environment. Its genetic code, its recipes for survival are just floating around. Now, another bacterium might come along. And some bacteria have the ability to just grab this free-floating DNA and pull it inside themselves. They can then stitch this new piece of DNA into their own genome. It's like finding a recipe card on the street, picking it up, and adding it to your own recipe book. If that recipe is for something useful, like how to resist an antibiotic, the bacterium that picked it up just got a major upgrade. This was a shocking discovery when scientists first saw it. They realized that the very essence of an organism, its DNA, wasn't locked away safely. It could be shared even after death. The second method is called transduction. This one involves a middleman, a virus. Specifically, a type of virus called a bacteriophage, which is a virus that infects bacteria. These viruses are like little syringes. They inject their own genetic material into a bacterium and hijack its machinery to make more viruses. During this process, when new viruses are being assembled, sometimes a mistake happens. A piece of the bacterium's own DNA gets accidentally packaged into a new virus particle instead of the virus's DNA. Now you have a virus carrying a chunk of bacterial DNA. This virus then goes off and infects another bacterium. But instead of injecting viral DNA, it injects the DNA from the previous host. The new bacterium receives a genetic gift from its neighbor delivered by a virus. It's like a mail carrier accidentally delivering a package to the wrong house, but the recipient decides to keep the contents anyway. This is a powerful way for genes to move around, because viruses are everywhere. The third, and maybe most fascinating, method is called conjugation. This is the closest thing bacteria have to sex. It's a direct cell-to-cell transfer of DNA. One bacterium grows a thin, tube-like bridge called a pilus. It extends this pilus and connects to another bacterium. It's like a handshake or building a temporary tunnel. Through this bridge, the donor bacterium can send a copy of a piece of its DNA to the recipient. Often, the DNA that gets shared is on a small circular piece of DNA called a plasmid. The plasmids are separate from the main bacterial chromosome. They're like little extra hard drives that carry bonus features. A single plasmid can carry genes for several useful traits, like resistance to multiple antibiotics. The donor bacterium copies the plasmid and sends the copy through the pilus. Now both bacteria have it, and the recipient can now do the same thing, passing the plasmid on to others. This process can happen very quickly, allowing a useful trait to spread through a bacterial population like wildfire. These three methods, transformation, transduction, and conjugation, are the workhorses of horizontal gene transfer in the bacterial world. They are constantly shuffling genes around, creating new combinations, and allowing bacteria to adapt at a stunning pace. So what are the consequences of all this gene swapping? One of the biggest and most direct impacts on us is antibiotic resistance. This is where horizontal gene transfer becomes a serious problem for human health. Imagine a single bacterium somewhere in a population that, through a random mutation, develops a gene that makes it resistant to an antibiotic, say penicillin. This gene might allow it to build an enzyme that breaks down the antibiotic molecule. When you take penicillin, all the other bacteria die, but this one resistant bacterium survives. It then starts to multiply. So far, this is just standard natural selection. But now, horizontal gene transfer kicks in. That resistant bacterium has its special gene on a plasmid. Through conjugation, it can connect to a nearby bacterium on even one from a completely different species and give it a copy of that plasmid. Now, there are two resistant bacteria. They each share it with two more. Then 4, then 8, then 16. The resistance spreads exponentially, far faster than if the bacteria had to just wait to reproduce vertically. Soon, you have a whole community of bacteria, a mix of different species that are all resistant to penicillin. And they didn't have to evolve it themselves, they just got it from a friend. This is why we see the rise of superbugs, bacteria that are resistant to multiple antibiotics. They've been collecting resistance genes through horizontal gene transfer, stockpiling them on plasmids, like a doomsday prepper stocks canned goods. It's a major reason why doctors are so concerned about the overuse of antibiotics. We are creating the perfect environment for this rapid sideways evolution to happen. But it's not all bad. Horizontal gene transfer is also a massive engine of innovation for life on Earth. It allows organisms to acquire completely new abilities in a single leap. A bacterium living in a toxic waste dump might evolve a gene to break down a specific pollutant. Through horizontal gene transfer, it can share that ability with other microbes in the area. Soon, the entire microbial community is working together to clean up the mess. Scientists have found bacteria that have acquired genes to digest oil, plastic, and pesticides. They didn't have these abilities ancestrally, they got them from the genetic marketplace. It could also lead to new ways of life. Think about photosynthesis, the ability to turn sunlight into energy. We associate it with plants, but some bacteria do it too. And horizontal gene transfer has moved entire sets of photosynthesis genes between different bacterial lineages. An organism that once lived in the dark could suddenly acquire the power to live off sunlight. Another example is the evolution of life in extreme environments. Bacteria that live in boiling hot springs or deep-sea hydrothermal vents need special proteins that can withstand the heat. Horizontal gene transfer allows them to share these heat resistance genes, enabling communities to thrive in places we once thought were lifeless. It's a way for life to quickly colonize new and challenging niches. This constant sharing of genes has forced scientists to rethink one of the most basic ideas in biology, the tree of life. The classic image, first drawn by Darwin, shows a single trunk at the bottom representing a common ancestor, which then branches out and out into the different species we see today. It's a neat, orderly picture of divergence. Vertical inheritance fits this model perfectly. But horizontal gene transfer messes it all up. If a bacterium can get a gene from a distant cousin, or even a completely unrelated species, then the branches of the tree are all connected by crosslinks. It's not a tree anymore, it's more like a tangled thicket or a web. Some scientists now talk about a web of life instead. The core of the tree is still there. Organisms are still related through vertical descent. But the horizontal transfers create a network of connections that overlays the tree, sharing innovations across different branches. This means that defining a species in bacteria is incredibly difficult. If two bacteria can freely exchange DNA, are they really separate species in the same way a lion and a tiger are? The lines get very blurry. And this isn't just a story about bacteria. While they are the champions of horizontal gene transfer, it happens elsewhere too. It even happens, to a lesser extent, in more complex organisms eukaryotes like us. Eukaryotic cells are the ones with a nucleus, the kind that make up plants, animals, and fungi. One of the most profound examples of HGT in our own deep history is the origin of our own cells. Eukaryotic cells contain tiny structures called mitochondria. These are the powerhouses of the cell. They generate our energy. The amazing thing is, mitochondria have their own DNA, separate from the DNA in our cell's nucleus. The leading theory, called endosymbiosis, suggests that long, long ago, an ancestral cell engulfed a bacterium but didn't digest it. Instead, they formed a partnership. The bacterium provided energy, and the host cell provided protection and nutrients. Over millions of years, most of the bacterium's genes were transferred from its own genome to the host cell's nuclear genome. This was a massive horizontal gene transfer event. The bacterium became the mitochondrion. A similar thing happened with chloroplasts, the structures in plant cells that perform photosynthesis. They were once free-living photosynthetic bacteria that were engulfed and became part of the plant cell. So, the very cells that make up our bodies are the result of an ancient, large-scale horizontal gene transfer. But what about more recent examples? It happens. Viruses are a major culprit. When certain viruses infect an animal or plant, they sometimes insert their own DNA into the host's genome. If this happens in a sperm or egg cell, that viral DNA can be passed down vertically to the next generation. It becomes a permanent part of that species' genetic code. If you look at the human genome, you'll find that a significant chunk of it, something like 8%, is made up of the remains of ancient viruses that infected our ancestors. These are called endogenous retroviruses. Most of them are just junk DNA now, fossilized viruses that no longer function. But some of them have been co-opted and now perform useful functions for us. For example, a gene that is essential for the formation of the placenta in mammals, the organ that connects a developing fetus to its mother, actually came from a virus. A viral gene, acquired through horizontal transfer millions of years ago, is now fundamental to how we reproduce. Plants get in on the action too. A type of bacteria called Agrobacterium is famous for its ability to transfer a piece of its own DNA into a plant's genome. It uses this to hijack the plant cell and force it to produce food for the bacteria. It's a natural genetic engineer. Humans have actually learned from this and now use a modified version of Agrobacterium as a tool in genetic engineering to insert new genes into crops. Some research suggests that this natural process has happened on its own in the wild. The sweet potato, for instance, naturally contains genes from Agrobacterium. They were transferred horizontally long ago and are now a stable part of the sweet potato's genome. Even animals can do it, though it seems to be much rarer than in microbes. One of the most spectacular examples is the green sea slug. This little creature eats algae, but it does more than just digest it. It steals the chloroplasts from the algae and incorporates them into its own cells. These stolen chloroplasts continue to photosynthesize, providing the slug with energy from sunlight. The slug can live for months without eating, just by sunbathing. For a long time, scientists wondered how the slug kept these chloroplasts alive, since the chloroplasts need certain proteins that are coded for in the algae's nuclear DNA. The amazing discovery was that the slug had genes for these proteins in its own genome. It had somehow acquired these genes from the algae through horizontal gene transfer. It didn't just steal the hardware, it stole the software to run it too. Another example that got a lot of attention involved tardigrades, also known as water bears. These microscopic animals are famous for being almost indestructible. They can survive being frozen, boiled, irradiated, and even the vacuum of space. The first analysis of their genome suggested that a huge portion of it, maybe up to a sixth, came from other organisms like bacteria, fungi, and plants via horizontal gene transfer. The idea was that their incredible toughness came from this collection of borrowed genes. However, later studies questioned this. They suggested the original findings might have been due to contamination of the samples. The scientific community is still debating exactly how much horizontal gene transfer happens in tardigrades, but it shows how this idea is now a serious consideration even when studying complex animals. Science is a process, and this is a perfect example of how ideas are tested, challenged, and refined. So how do scientists even figure out that a gene has been transferred horizontally? It's like being a detective looking at a genome. One of the main clues is when a gene just looks out of place. You can build a family tree for a group of organisms based on their overall DNA similarity. Then you can build a separate family tree for a specific gene. If the gene's family tree doesn't match the organism's family tree, that's a red flag. For example, you might find a gene in a species of bacteria whose closest relative is a gene from a fungus. It's highly unlikely they inherited it from a common ancestor millions of years ago. It's much more likely that the bacterium got the gene from a fungus somehow, somewhere along the way. Another clue is the composition of the DNA itself. DNA is made of 4 bases A, T, C, and G. Different organisms have different tendencies in their DNA. Some have a higher percentage of G and C bases, what's called a high GC content. Others have a lower GC content. If you are looking at the genome of a bacterium with low GC content and you suddenly find a stretch of DNA with very high GC content, it looks suspicious. It's like finding a paragraph written in a different language in the middle of a book. It suggests that this piece of DNA came from somewhere else. It's a foreign import. Using these and other computational tools, scientists can scan entire genomes and identify genes that are likely to have been acquired horizontally. It has revealed that these transfers are not just oddities, they are a widespread and powerful force. What does all this mean for our big picture view of life? It means that life is more fluid and more connected than we ever imagined. Organisms are not just isolated islands of genetic information evolving on their own. They are part of a global network, a genetic commons where DNA can be shared and exchanged. This is especially true for the microbial world, which makes up the vast majority of life on Earth. A gene that evolves in one corner of the planet can, in theory, spread to a completely different environment and a different type of organism. This has practical implications beyond antibiotic resistance. In biotechnology, we are learning to harness the tools of horizontal gene transfer. As mentioned, the way we create many genetically modified plants is by using the agrobacterium system, a natural gene transfer machine. The new gene editing technology CRISPR, which has revolutionized genetics, is actually based on a bacterial immune system. It's a system that bacteria evolved to protect themselves from invading DNA, like that from viruses. They store little snippets of viral DNA to recognize and chop up future invaders. It's an immune system designed to fight against a form of horizontal gene transfer. We have now repurposed that bacterial defense system into a tool for editing the genomes of all kinds of organisms, including humans. Understanding horizontal gene transfer is also crucial for understanding disease. It's not just about resistance. Some bacteria are harmless until they acquire a set of genes called a pathogenicity island from a more dangerous bacterium. These islands contain genes for toxins or other things that allow the bacterium to cause disease. A harmless gut bacterium could, in theory, become a serious pathogen in a single transfer event. In the end, horizontal gene transfer is a fundamental aspect of evolution. For a long time, we focused on the slow, gradual change caused by random mutations and passed down vertically. That is still incredibly important, but horizontal gene transfer adds a new dimension. It allows for rapid, dramatic leaps in evolution. It's a source of instant innovation. It's what makes the microbial world so dynamic and resilient. It complicates our simple stories about evolution, but it also makes the story of life richer and much more interesting. It shows us that life isn't always a competition between isolated individuals. Sometimes it's about cooperation and sharing. The boundaries between species are not as solid as they appear. They are porous, and through the tiny holes, the very instructions for life itself are constantly flowing. It's a reminder that all life is connected, not just by a distant, ancient tree, but by a present, active, and ever-changing web.